Welcome back, everyone. Hopefully exam 1 went well. We're fixing to move into the material for exam 2, which is traditionally the most difficult exam. So if you had difficulties or questions about exam 1, talk to me as soon as possible so we can get you pointed in the right direction, because this is the make or break exam typically in terms of your final average. With that said, let's get started. So chapter 5 is all about the cell cycle, which the cell cycle is how you regulate and control cell division, which is the process that allows a single fertilized egg to grow into an adult with a few trillion cells. Your somatic cells are the cells of your body, and they continue to undergo division even into adulthood, replacing things like your blood or your skin. You're constantly sloughing off skin cells because this outer layer that you see is dead. Which is why exfoliation kind of makes you look a little more vibrant, because you've literally just buffed off a layer of dead tissue. And your blood cells are constantly being broken down and replaced because red blood cells do not have a nucleus, so their lifespan is only about 120 days in a healthy individual. So you have to constantly replace those in order to carry your oxygen and so on and so forth. Sometimes, though, it is necessary to kill cells, and that process is called apoptosis, or under normal controlled circumstances. Programmed cell death is apoptosis. You have plenty of other ways of killing cells that do not follow this pattern. Certainly don't want to get into necrosis, because that's something else, just killing all of your tissues. But apoptosis is useful for decreasing the number of cells, and occurs when you need to remove unwanted tissue, or if you notice that a cell is proceeding to the cell cycle incorrectly in an unregulated fashion. It's probably cancerous, so you want to induce apoptosis in that cell immediately. In humans, we use it to remove the webbing between our fingers and toes before we're born, and for tadpoles, they lose their tail as they develop into frogs in a controlled manner. So the cell cycle is the sequence of events that takes place during cellular division. And we have two main phases for that: interphase and the mitotic stage. The mitotic stage is broken down into mitosis and cytokinesis, which we will break down each of those categories in more detail as we go through. So starting with interphase, G1, you double the organelles and accumulate all the material you need for DNA synthesis. For S phase, you grow and synthesize DNA. Each chromosome consists of one DNA molecule called a chromatid. After DNA replication, each chromosome consists of two sister chromatids. So you go from one, you copy it, and you bind them together here at the centromere so that you've got everything kind of organized and together. And then once you've done this, then you move into G2, which synthesizes all of the proteins necessary for cell division. Now, eukaryotes have a complex system for regulating the cell cycle. We have checkpoints at multiple points along the cell cycle that ensure that everything up to that point has been done correctly, and then that will allow the cell to proceed. If you fail one of these checkpoints, then there's a very high likelihood that that cell will undergo apoptosis, because a damaged cell should not be dividing, because if it is, there's a high likelihood it's going to be cancerous. So you have your three main checkpoints here are at G1, G2, and M. At G1, if your DNA is damaged, you stop the division right then and there. G2, if you did not replicate all of your DNA, stop it right there. And then at M phase, if the chromosomes are not properly distributed, and we'll talk more about what happens when a chromosome is not distributed correctly in a minute, but spoilers, trisomy and monosomies will result from that. So mitosis, through mitosis, we have to maintain the number of chromosomes in each cell. Each species has its own characteristic number of chromosomes. So for humans, it's 46. You've got 23 pairs, 46 chromosomes and 23 pairs. 44 are autosomal, 2 are sex chromosomes. For dogs, it's 76. And there are some plants, like certain wheats and things, that have hundreds of chromosomes because they seem to tolerate duplicating them better than we do. It's just a weird plant thing, nothing you really have to worry about for this course. For your somatic cells are all diploid, meaning you have 2 pairs of each chromosome, except your sex cells, you just have 1 of each of them. And then for your haploid cells, you take that number, divide it in half, so you're 23 somatics, and then 1 member of your of each pair of your sex chromosomes. And then when those are fertilized, it restores the haploid number, so then you end up back with your 46 and 2, or I'm sorry, your 44 and 2 for 46 in total. During mitosis, you have nuclear division in which the chromosome number stays constant. So basically, you break the nucleus of the cell down, you copy your DNA, and then each resulting daughter cell is going to get 1/2 of that, because again, we talked about here duplicating your sister chromatids, and then 1/2 goes to each daughter. So the definition for mitosis here is the division of the nucleus that follows interphase. And you're going to see this PMAT a lot. It's prophase, metaphase, anaphase, telophase. Sometimes you'll hear this, there's an intermediate step that they'll call prometaphase. Sometimes it's broken down into prophase and late prophase. Just for our purposes in this course, just PMAT. Cytokinesis is the division of the cytoplasm that follows mitosis, and you get 2 daughter cells that are identical to the mother. So you have mitosis, cytokinesis. So breaking this down a little bit more, you've got prophase. The nuclear membrane disappears, the centrosomes migrate, the spindle fiber appears, and then the chromatin condenses and chromosomes become visible. And each is composed of 2 sister chromatids, and then the centromere is attached to the spindle fibers. So if we can zoom in a little bit here without breaking the computer. And some days that's a big if. Okay, here we go. So early prophase, you're breaking down the nuclear membrane, you're replicating your DNA. During prophase, the middle of prophase, you have all of your chromosomes condensed down to where, and they're duplicated and bound at the centromere with your 2 sister chromatids together. Late prophase, then you have the spindle fibers here attaching, and they're going to start lining up the DNA down the middle so that you can equally divide it for the 2 daughters. So metaphase, you've got the spindle fully formed and the chromosomes line up at the metaphase plate right here. Anaphase, you pull the sister chromatids apart, which are now referred to as chromosomes, and they're moved to the opposite poles by the fibers. And this is accomplished by shortening the fibers once you have attached them to the chromosomes. And then telophase, you form this cleft in the cytoplasm, and it will physically start dividing the cell into two, and you have your 2 separate nuclei with your DNA and copies of all of your organelles and everything here ready to go. And this is referred to as the cleavage furrow, and it has this little contractile ring, right? It literally, like the ring, literally just pinches the 2 in half. Which you can see diagrammed, the whole process diagrammed here. This diagram's a little bit simplistic, but it's pretty good for getting the right idea in your head. It's a little different in plants because you have a cell wall in plants. So instead of this contractile ring and the cleavage furrow, you have the formation of a cell plate in between the 2, which ends up producing a second, cell wall between the 2 daughter cells. Okay, so mitosis maintains the chromosome number. Meiosis reduces the chromosome number, and meiosis is the process by which you produce gametes, or sex cells. And this occurs in all sexual reproducing organisms, which, yes, there are plenty of organisms that reproduce asexually, and that has certain benefits, but the primary benefit of sexual reproduction is a genetic reshuffling, so that your offspring are genetically different than their parents, and this produces a certain degree of diversity within the population, so that you're not all susceptible to the same things. I don't know if you all have ever noticed this, but if you get banana flavoring, it literally tastes nothing like a banana. And that's because the bananas we have now are not the same as the bananas we have when that flavoring was made. We have what is called a Cavendish. And when banana flavoring was originally made, we had a banana called the Michelle Grove. And the thing is, bananas have been selectively bred for so long to reduce the number of seeds in them and the amount of it that is edible, that the ones that we grow are all clones now. They cannot reproduce on their own. And because all of the Michelle Groves were genetically identical, there was a particular form of fungal blight that was able to infect them, and because one was susceptible, they were all susceptible, and it wiped them out. Fortunately, we were able to use a different variety called the Cavendish and replenish our banana crops. But there is a new form of the blight that has emerged that can infect the Cavendish. So maybe in 10 years we'll have another strain of cultivated banana that everyone regularly consumes. I don't know, something like a plantain or something. It'd be interesting to see. But that aside, this genetic variation is produced in two ways: crossing over and independent assortment, and we'll get into those in a little more detail as we go through here. But meiosis begins with one diploid parent cell, and it requires two cell divisions, meiosis 1 and meiosis 2, which results in 4 haploid daughter cells. The thing to keep in mind here is that meiosis 1 is essentially identical to mitosis. It is really meiosis 2 where things get changed up a bit. And also, do your best to keep mitosis and meiosis separate in your brain, because yes, they are very similar and they sound very similar, but there are key differences between them that you're going to have to discriminate between on the exam. So, that said, let's proceed. So basically what you end up doing here is you do your meiosis 1, and then during the meiosis 2, you move the pairs of chromosomes called homologs, which are inherited from each parent that line up at the equator, and then you separate them into the daughter cells, and each one receives a single member of the pair, and these cells are now haploid cells. So you did basically everything you did in mitosis for meiosis 1, then you do it again for meiosis 2, except you don't recopy the DNA a second time to produce your haploid cells. So your first division here, you've got prophase 1, metaphase 1, anaphase 1, telophase 1. Just like mitosis, so far so good. Everything happens pretty much like we did before. The key difference here is prophase 1, you have this crossing over event, which allows DNA to be exchanged between these pairs of chromosomes. So you end up with a little bit of shuffling here. Say the red copy was from mom and the blue copy was from dad. Well, now you have switched bits of DNA so that you've got some traits on this one from one parent and some from the other, instead of them just being straight up copies of their parents. So that's your first set, or your first infusion of variation right there. And then during metaphase 1, you have this independent assortment. So You've copied everything, you send them off into the two daughter cells, and then, okay, well, we don't have a good picture right here, but you can see that not every chromosome, like you don't have to have the chromosomes from one parent go into one cell and one parent go into the other. They can, but frequently you have a mixture of the two. So you have chromosomes that have mixtures of different traits from both parents, and then you have cells that are produced that have combinations of chromosomes from both parents. So if memory serves, and I didn't do the math on this personally, but the math has been done. If you have two individuals, you're supposed to be able to have a few billion offspring before they have two genetically identical children, barring identical twins. And so that gives you some idea of how much variation is produced in these two steps. Okay, during anaphase 1, your homologous chromosomes separate and pour towards opposite poles. Telophase 1 may or may not occur at the end of meiosis 1. The nuclear envelope reforms and nucleoli reappear. Cytokinesis may occur. Two haploid daughter cells are then formed, and then you have interkinesis. Now here's the main thing. This is not like interphase because you're not replicating your DNA. It's just a gap between meiosis 1 and 2. So then you have your second round of cell division. You have prophase 2 and metaphase 2. And the key thing here is in prophase 2, your duplicated chromatids are attached to the spindles, and then in metaphase 2, they line up at the metaphase plate and are separated. So the result, and you have anaphase 2 and telophase 2, which goes just like it did in the previous steps. But if you zoom in on this chart without breaking the computer, hopefully. So meiosis 2, the chromosomes are color-coded here so you can see like which ones go where for your independent assortment and your crossing over. So you've got crossing over going on here, and then you line them up on the plate. In prophase 2, you line them up on the plate in metaphase 1. Anaphase 2, you separate them out, and then you go straight into telophase 2, you go into telophase 2, where you've separated everything out, and now you've got your two, you form the cleavage furrow. You've got your two daughter chromosomes, and you can see, just based on the color here, how different these are compared to the original cells. And that's kind of the point. So the takeaway here is that meiosis produces 4 haploid cells that are genetically unique, and mitosis produces 2 diploid cells that are genetically identical. The other key things here is that meiosis is used for producing gametes, mitosis is used for producing somatic cells, and the entire point of meiosis is producing genetic variation. And that's essential, and that genetic variation is essentially what makes us all unique individuals and prevents us from being just clones of our parents, essentially. And this whole thing is kind of summed up here in what they're calling the human life cycle. You've got mitosis takes you from a zygote to a baby. The baby then continues mitosis until it reaches adulthood. Then you would use meiosis to produce your gametes, and then the gametes then fuse to fertilize and produce a new zygote, and so on and so forth. Few things that they're not quite relaying here properly. A female is actually born with all of the eggs she ever has. Those are produced in utero, and that's it. You've got them. they're produced in utero in the ovaries, and they're just kind of stored there until they're needed. They don't make any more, which is one of the reasons why over time the risk of say autism or Down goes up with older mothers, because those cells have sat around for decades and accumulated more damage over time and are more likely to have errors in this process. Males, on the other hand, constantly produce new sperm. The male is essentially the purpose of the male in sexually reproducing species is to produce genetic diversity. They're constantly shuffling and shuffling their DNA in their gametes and producing new combinations of traits. Females are more they're much more stable than the males in this regard. And the other thing that I don't think it was specifically mentioned is that the male is actually the one that is responsible for the biological sex of the offspring. A female has 2 X chromosomes, so the egg will always have your 23 somatic and 1 X chromosome. The male can produce sperm that have the 23 somatic and an X, and the 23 somatic and a Y chromosome. And so depending on which one of them actually ends up fertilizing, that determines the biological sex of the offspring. With that being said, that concludes Chapter 5, and we'll see you next time.